So, welcome back. Today I am going to talk a bit about mitosis and cell cycling in general. Right? And so, this is the process that allows our cells to make new cells. Now, one of the things about cells is that all cells come from pre-existing cells. That's one of the tenets of cell theory. Right? You can't just like make a cell out of nothing. All the cells in our body came from other cells. At one point in time, there was one original cell, and we've all have evolved from that one original cell. Okay? So we cannot have spontaneous generation of cells. In order to make new cells, we have to take a pre-existing cell and split it into two cells. Okay? And that process is mitosis. And that's the name we use to describe that splitting. And there is mitosis, which is a step in the general cell cycle. And so our cells have their own little life cycle, its own little circle of existence. Right? It's kind of born, and then it kind of makes babies, or it could die. Right? So those are its options. It can turn into new cells, or it can die. Right? And the cell cycle are the steps that prepare this cell for splitting into two new cells. All right. So the beginning of the cell cycle is the end of the cell cycle. So mitosis is both the beginning and the end. So mitosis is making these two new baby cells. When the cells first come out into the world, they enter a resting phase called G0. And now G0 is where these cells are just chilling and doing their cell thing. They're the perfect size to do their job. They have all the things inside of them that they need to do their job, and they're just chilling, doing their job. So if this is a liver cell, it's chilling, doing liver things. If it's an eyeball cell, it's chilling, doing eyeball things. So this is when it's doing its job. Now, at some point in its life, the events around it might trigger it to say, hey, buddy, it's time that you divide. You've lived long enough. You've got to make yourself into two new baby cells. And so the process of cell cycling begins. And so this cell is moved from G0 into G1. Okay, growth phase 1. You also see this referred to as gap phase 1. I don't like it because like growth phase describes what it's doing. It's growing. Okay? So growth phase 1 is what we're going with. Okay? And what it's doing is it's getting bigger. It's adding more cytoplasm. It's adding more copies of its organelles. Right? And so when it was in G0, it had the right amount of organelles to do its job. If this one cell is going to become two cells, though, it needs two cells worth of organelles. Okay? So we're making extra copies of our organelles in this phase. Right? And so in this little model, the smiley face represents organelles. Okay? This little circle here represents DNA. So after G1, you see that we now have two smiley faces, signifying that we have copies of our organelles. We still only have one circle though. Okay? Now we're moving into our next phase, which is S phase, which is synthesis phase. Okay? Now this is the synthesis of DNA. If I'm going to make two cells out of one cell, I'm going to need two copies of that cell's DNA. Okay? And S phase is what's happening here to make that second copy. A process called DNA replication is occurring, and it's literally making a photocopy of the DNA that exists. So I have one full copy before S phase, and by the end of S phase, I have two full copies of this organism's DNA chilling inside this cell. Okay? So by the end of S phase, we had chonky cell, we got two copies of DNA, we got multiple copies of our organelles. Okay? We're almost ready to divide. We just need to grow a little bit more, get a little chunkier, right? And then we're going to add these little structures called centrosomes. Okay? So these centrosomes are maturing in this next phase, which is called growth phase 2. Okay? So G2 happens immediately prior to cell division itself. Right? So to recap, we have M phase, G0, G1, S, G2, and then we're back into M phase. So here's our cyclical model, right? M, G0, G1, S, G2, back into M. Okay? So that's why the beginning is also the end. Right? So we are here now with our chunky cell, has everything it needs to become two different cells. We just now need to divide it, split it in half. And so in order to split it in half, first things first, we have to make sure that each half gets the right set of DNA. Okay, so we now have multiple different variations of our chromosomes. We have two copies of each of those chromosomes, and we need both cells that are being made to have a full set of that DNA. Okay, I can't accidentally put one chromosome over here and four chromosomes over there. Not going to work. It has to be a nice even split, okay, with all of the chromosomes making it. And so mitosis itself is the process of dividing the DNA evenly. You have to make sure that both sides get the full set of DNA. So both baby cells end up with this full DNA. So by the end of mitosis, I now have two new cells with the right amount of organelles and one set of DNA. And these are going to go back into G0, and the whole storyline is going to start all over again. Okay. So that's the general cell cycle. Now we're going to look specifically at just mitosis, the M phase of mitotic cell cycling. So as I said, mitosis is the process that separates the DNA evenly into these two new daughter cells. And it occurs in a few steps. Okay? The steps as a list are prophase, metaphase, anaphase, and telophase. Those are the steps of mitosis. And we abbreviate it as PMAT, okay? As in a mat that one might urinate upon, a PMAT. Okay? So prophase, its job is to break down the nucleus and condense the chromosomes. So while the cell is doing its job, its DNA are these long, thin strands wrapped around some proteins called histones, right? It's what I refer to as spaghetti DNA. Okay? If you imagine a plate of spaghetti, that's what the DNA looks like when we're in G0 and just chilling and when we move through, okay? Up until prophase, this DNA is shaped like spaghetti. Okay. Prophase is breaking the nucleus down so that the DNA can float around the cell. Okay. And it's also condensing the DNA into these tight mitotic chromosome packages that I refer to as ZT DNA. Okay. So we have a plate of spaghetti versus a plate of ZT. Okay. So spaghetti is all tangly, right? If you imagine trying to separate spaghetti strands without breaking into them, it's hard. Okay. But if I give you a plate of ziti, right, it's really easy to pick one ziti up and put it on one side, pick another ziti up and put it on the other side, right? So after prophase, we have ziti DNA. Okay. Metaphase's job is to line those organized chromosomes up down the middle of the cell. Okay. So the chromosomes are just going to make one pretty little line, nice and straight down the middle. Okay. The order in which they line up doesn't matter at all. Not one bit, okay? No matter. They just need to make sure they're in a nice straight queue, okay? As long as in that straight queue, everything's going to be all right. After metaphase, we go into anaphase, okay? So here we have anaphase. Okay? And anaphase separates our copied DNA into two pieces, okay? And so when my DNA copies, the copy itself is stuck to its original piece, right? So if this is my original chromosome, during the copying phase, I make an exact copy and they're stuck together like this down the middle. Okay. Anaphase is taking these two exact copies and just splitting them apart. Okay. And moving them to the other sides of the cell. And so in this way, both sides get exactly the same amount of DNA and they get all the chromosomes, right? Because it's just copies being ripped in half and being pulled opposite directions, right? So it's like if you had me and then you copied me, right? And you put one side of me over there and one of me over there, both ends of the room get a big, beautiful me, right? So nobody misses out because that would be just too much of a shame to miss out on this, right? So that is what is going on in anaphase. We're separating the copies of the DNA to both sides. In telophase, telophase, we are rebuilding the nucleus. Okay? So the DNA goes back to being spaghetti form, and we wrap them back up in a nuclear envelope. Okay? That is mitosis. We're done. The DNA is separated evenly. The cells still need to be split up into two. That last step of actually ripping the cells into two separate new daughter cells is called cytokinesis. Okay? Kinesis as a suffix is referring to movement. Okay? Kinesis means movement. Okay? And cyto as a prefix is referring to the cytoplasm. Okay? We're cutting the cytoplasm in two and making these two new cells. Okay? So that wraps up the whole process of cell division, is cytokinesis as a word. Okay? Divides the cell into two. Alrighty. And so, as you can see, this whole process makes two clones of the parent cell, right? So whatever kind of cell I started with, I now have two identical clones to that. So if I started with an eyeball cell, I now have two eyeball cells. If I started with a liver cell, I now have two liver cells. The function of doing this is for growth and repair of the organism. Okay? So when I was a wee little baby, I had way less cells than I have now. Right? I needed more cells to get this big and beautiful. Right? And so my baby cells did a lot of mitosis. It's mitosis after mitosis after mitosis after mitosis. Boom. And 25 years later, I was this guy. And then 10 years later, now I'm old. Right? But anyways, so my mitosis of growth stopped a while ago because I got old. Yeah. That's how I grew into this full adult form, is through the process of mitosis. Now that I'm old and I don't need to grow anymore, right? I'm not growing ever again. My body still needs to do mitosis because my cells can become damaged. Right? Just the process of living might damage them. They get old, they get beat down, they die. Right? And so I need to replace those damaged cells with new, fresh cells. Okay? I could be injured, and those injured cells need to be replaced, right? So whatever needs to be repaired is also done through mitosis. Okay? So if I got like stabbed in the eyeball, right? Those damaged cells are dead. They need to be replaced with new cells. So the cells next to them will do mitosis, and they'll replace the damaged cells, and my eyeball will be fixed so I can still see. All right? Yeah. So that's it. Mitosis. One cell turns into two cells so that we can grow and repair. Ta-da! Have a great day. Bye.